We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
So we're now set to begin our live stream Q&A session where the speakers in our symposium will answer a selection of thought-provoking questions from the audience. With that, I would like to invite our speakers back into the Zoom room. I just want to ask one timely question, start off with one question for Chip Schooley. So this is from one of the students in the course. UCSD is currently planning and implementing its Back to Learn program, large-scale campus-wide testing for novel coronaviruses in the fall, in what seems to be a test, trace, isolate, support approach. It is estimated that by monitoring 60 to 90% of the campus population, greater than 90% chance of detecting virus spread at a time when less than 10 of the 60,000 members of the campus population are virus shedding can be achieved. The campus, however, is not a closed system. And how often would retesting have to occur for such a strategy to be successful? Without a treatment or vaccine in sight by the fall, would we be taking a risk on at-risk individuals and risking some fatality for the sake of a greater good?
Let's start with the question about how often we would have to test. Based on the modeling that Natasha Martin and Victor Diagotola have done, the testing would have to be done once a month. So it would be a lot of testing, there's no question about it. All of this has to be taken in the context of how much activity there is in the community. And if there's a lot of viral activity in the community, we won't be back at school. There's no question about that. And I'm concerned about the trends we've seen over the last several weeks, in fact, about both around the country and frankly here in Southern California. But the goal of this is if things are quiet enough to get back, that we can prevent there from being an outbreak that we don't know about. So it's really more of an attempt to, if the society is getting back to work, to have us be one of the safer places to be so that we know what's going on before something happens. It's not an attempt to have us be reckless and get back when others can't. It's an effort to be able to be vigilant when we do get back to mitigate any damage that might occur from spread of the virus.
Okay, thank you. Victor?
Okay, I'm gonna go with my next question from CARTA student Jason Adams, and this is directed towards Manuela Rafatellu. And the question is, in what ways does intra-individual variation in the human gut microbiota influence Salmonella, or I guess any other enteric pathogen, if they're better examples, in terms of disease severity?
I think, Jason, you ask very important questions about individual variability to susceptibility to gastrointestinal infection. And what we know is indeed that the gut microbiota plays an important role in providing colonization resistance to pathogens. For example, the use of antibiotics that definitely provides an increased susceptibility to enteric pathogens like Salmonella. And there is definitely individual variability that we don't completely understand yet. But what I can tell you is that there are some organisms that are known to provide colonization resistance to Salmonella. For example, there are Clostridia that produce short chain fatty acids that are important modulator of intestinal immunity and epithelial cell homeostasis. And these are well known to provide colonization resistance and to help maintaining the gut well balanced. And in fact, one of the consequences of inflammation induced by Salmonella is to kill those Clostridia with the goal of providing a niche for the pathogen. But yes, definitely the gut microbiome and individual variations are important in determining susceptibility or resistance to enteric pathogens in general.
Thank you, Manuela. Back to Elizabeth for the next question.
Okay, this is a question from Janelle Giller, and it's for Mandy Lewis. Thank you so much for the informational presentation. My question is, what does data show regarding the impact of oral probiotic supplements on the vaginal microbiome? And can it shift the microbiome to a lactobacillus dominant microbiome?
Can probiotics shift the microbiome to a lactobacillus dominant microbiome? One of the problems with the current commercially available probiotics is that they are intestinal lactobacilli mainly. There are a number of studies going on with vaginal lactobacilli, which seem to have a much more prominent effect on women's health, including the ability to colonize the vagina, whereas the intestinal strains are incapable of doing that.
Okay, thanks, Mandy. I guess I'll take the next question, and this will be directed to Sujan. And the question is, why isn't ADE a problem for infection with other viruses aside from dengue, or is it?
This is the question related to ADE is a really important one, not just in the context of dengue infections, folivirus infections, but we think it may be applicable to other infectious diseases where you have this immunological cross-reactivity. In that this cross-reactivity could be good, and Susan Keck just gave a beautiful presentation on how Jenner used it to his advantage to create this first concept of vaccination, from giving cowpox to eradicate smallpox now. And so the cross-reactivity is good in that case. And unfortunately, with diseases such as dengue, we're finding out that this cross-reactivity, besides being good, can also play a pathogenic role, a bad role. And instead of providing this cross-protective immunity, you could actually come down with severe form of these different viral infections. And in the current time, in trying to bring our focus back to COVID, in fact, there are several investigators in the coronavirus field who are looking into the dengue field, what we know about ADE. And I'm thinking perhaps there may be issues related to ADE. For instance, there are young, healthy people who are coming down with severe coronavirus, severe COVID. It's not just a disease that causes severe disease only in the elderly or with comorbidities. And one of the hypotheses surrounding why these young, healthy individuals are coming down with severe COVID disease may be related to ADE due to these people's exposure to these common cold-causing coronaviruses, which are mild, but there is enough cross-reactive immunity that you can imagine under certain conditions in certain people when that antibody response, or it could even be T-cell response, is no longer good enough to protect, but instead now causing the more severe form of infection. So we think, despite the field taking multiple years, as I explained in my talk, over 50 years to really understand this concept of ADE using animal models and using epidemiologic field studies spanning multiple decades, multiple countries, we can apply what we've learned from the dengue Zika field and try to see how we can develop better vaccines against the coronaviruses, in particular against COVID. So we think this concept of 80 is really important, and there is no reason why it has to be specific for the flavivirus field.
The next question I have is from Gerata, and the question is, and maybe this is Sue can answer this best. What is the best guess about the possibility that no vaccine will actually be possible for COVID-19 in the long term?
What's the possibility? Well, I mean, I guess in history, we've had our share of challenges with certain pathogens, from HIV to malaria, and TB being very notable vaccines that have not led to the successes that one would hope. My honest feeling is that this virus, COVID-19, SARS-CoV-2, is not going to be as much of a challenge. I think in part because it's not one of these shapeshifters like we see with HIV, for instance, and other pathogens that do have ways of evading by changing the antigenic profile that the pathogen has. And also, we already have evidence that the antibodies that are being produced can be protective and neutralizing. So there are some viruses for which it is more difficult to develop neutralizing antibodies, particularly HIV. So I feel like this virus isn't holding those kinds of properties that have been historically more challenging to deal with. But of course, it does have evasive features. We know it's changing the interferon response very profoundly. We know that the pathology that is causing the death is due to over-exuberant immune responses. So those, understanding the nature by which this virus is leading to those changes is going to be important. But just my immunological gut tells me that we are going to develop a vaccine that will be protective. And I honestly think what's going to be important is that the vaccines are being currently tested, that we focus not only on the spike protein, which is what people are, but consider the other proteins in the virus as well. I'm a little bit nervous about the vaccines that just focus on the spike protein in some ways for the antibody-mediated enhancement that Sujan just was talking about. I think that could be a complicating factor of vaccines that just focus on the S protein, but that's just my opinion.
Thank you.
Okay, I'm going to pose the next question actually to Nisi Varki. And this came up during the course of all the presentations from a listener. Given that nearly every talk, including my own, I guess, talked about sialic acid, why is this such a common molecule for infectious disease pathogenesis?
Yeah, hi, thank you. Good question. Well, as I showed in one of my slides, and many of you actually showed the slides of the cell surface being decorated with glycans, glycoproteins and glycolipids. And at the very tip of all of these glycans is the sugar 9-carbon sialic acid. So the sialic acid's at the very tip of all the glycans, at the very edge of the sugar coating of each cell. So it's there available, and that's what most of the viruses and pathogens see as they're traveling by, either in the bloodstream or wherever. So these sialic acids not only interact with pathogens, they also interact with circulating cancer cells, platelets, immune cells. So they are really, so even in, I think, yeah, the MERS virus was, even though everyone was talking about the protein-protein interaction, there was a sialic acid interaction that was discovered. So like the human influenza virus recognizes the sialic acid, which is used to bind and invade, and you can inhibit that using Tamiflu. So we're hoping that something like that is similar is found, the interaction with the other viruses as well, and that would help with the treatment.
Thanks, Nisi. I guess it's true that the sialic acid receptor SIG-like family in humans is one of the most rapidly evolving regions of the human genome. Does that, I think, lend credence to the importance in evolution?
Right. So that's what piqued our interest early on, that this rapidly evolving Siglex, which is the sialic acid recognizing receptors, have been associated with a lot of the evolutionary events that have happened over the centuries. Yeah. So some of the Siglex, for example, are present on cancer cells, and that might influence the way the cancer cells invade and metastasize as well.
Thank you.
Okay. I have a question for Victor, and this one comes from Pascal. And the question is there evidence that group B strep was a major problem long before animal husbandry?
That's a good question. I don't think we have the historical data on ancient evolution, but we do have a lot of evidence that there are human-specific features of group B strep infection and immunity. For example, group B strep has molecules that are virulence determinants, which target specifically human IgA, but not IgA from closely related species and the experimental animals, only target human factor H, and not other complement regulatory proteins. The most other important medical incidence of GBS disease other than humans is in mastitis in cows, but those strains seem very distantly related from humans, and there's not a lot of evidence of exchange. However, pets like dogs and cats can rarely get disease from their human owners. And there are aquatic forms of group B strep in marine mammals and in fish, which do seem to have the potential for transmission back and forth and share virulence factors and genetic profiles with humans. So perhaps the advent of fishing and aquaculture as a subset of agriculture may have been an accelerator. All these differences, though, pose a great challenge to modeling of group A strep and other, pathogens like group B strep, group A strep, Staph aureus in animals, mice, rabbits, because they have such unique features. Some of the toxins that drive the pathogenesis in humans don't target the cells in the experimental animals. The superantigens, which can give you toxic shock syndrome, don't tend to react and produce the clonal expansion of the T cells in the experimental animals, which makes therapeutic development and vaccine development challenging because you worry that a candidate therapeutic or vaccine that performs best in animals may not be the one that ultimately performs best in humans when you go to the large phase 2 and phase 3 trials. I will have a question here actually for Elizabeth, right back at you. And this, let me find out who this question was from, because it was a really thoughtful one. This is from a CARTA student named Nico. Why did artemisinin resistance of Plasmodium only develop once it was used in its synthetic form and not during the thousands of years before, when it was used as Artemisia annua, sweet wormwood, as a natural drug? Does such knowledge affect the development of new antimalarial drugs in labs like your own?
Yeah, yes, and I would say that this is probably a question, the streetlight effect as well. It's actually very hard to test for artemisinin resistance. It's actually difficult to test for most resistance to most antimalarial drugs because it's mostly just correlation with molecular markers. Because it's very unlike, testing to see if strep are resistant to ampicillin or whatever antibiotic you're using, where you can easily culture the bacteria out from your bloodstream and then put them on a plate with a filter disc. Taking a parasite out, especially in an area with poor resources, and then taking them into cell culture and then testing them after a few days to see if they respond to a drug is actually incredibly difficult. I would say there probably was resistance, but we just weren't able to measure it because we were only looking, where there was light.
Okay, thanks, Elizabeth.
So this is a question for Chip, and it's: to what extent might COVID be a mild childhood disease and a more severe in adults, and would change in the immune system during adolescence possibly account for this?
That's also a great question, and there does seem to be quite a bit of difference in the frequency with which younger people become ill when they get infected compared to older people. It's also true that people who are younger tend to shed less virus during the course of their illness. Now, it's hard to separate those two out because, in general, the more severely ill you are with this disease, the higher the titers of the virus. So they go kind of hand in hand. One of the curious things that may also be an issue with vaccine development is that in the vaccine studies done in animal models, younger animals tend to be much more responsive to vaccines than older animals. And it may turn out that we will have to have different strategies for older people. And by that I mean people in their 30s, not just people who are slightly older than Victor and I. And when you think about that, you might have to have different either vaccines in general or different vaccination regimens, different vaccine doses, different adjuvants to be able to sufficiently stimulate the adult immune response for lasting immunity. It's also true that immunity to this class of viruses drops more rapidly than, say, influenza and some of the other viruses we encounter. And that may be one of the reasons why this virus, which evolves more slowly from the molecular perspective in populations, can still come back and cause recurrent waves of infection through the population at 3- and 4-year intervals. So I think we have a lot to learn about differences in the immune response that occur in this virus compared to older people. We still don't understand the recently described syndrome that we're seeing in kids, which is a hyperinflammatory multi-organ syndrome, very much like Kawasaki's disease, that we only have recognized in the last several weeks, and only in Europe and in the US. Was not seen in tens of thousands of cases in China and in the East. I'm glad we have Dr. Nazay here to explain to us the reason for this. So, Victor, what are your thoughts about this? Do you think?
Well, yeah, so as a pediatric infectious disease doctor, we would see a lot of patients with the syndrome known as Kawasaki disease, of which there is some overlap in the clinical features, as well as toxic shock syndrome, which could complicate staph or strep infections. I think this clearly would suggest that there are environmental cofactors or immunogenetic predispositions, which have been well documented in Kawasaki disease, for example. And another factor I wonder is whether there are special aspects of immune senescence as we age, or just the cumulative damage that occurs to our lungs living in a world that is not pristine but polluted, that are below clinical detection until they get a life-threatening challenge like COVID-19 pneumonia, and that children just have not accrued that amount of change over time, which could be interfering with normal epithelial immune responses. It's also possible maybe that the virus has a completely different spread and is more of a systemic viremic disease in children associated with minimal immune response in the majority of patients, but then the potential for this delayed immune-mediated disease in the unfortunate few. There's quite a few other diseases like Epstein-Barr virus or poliovirus, where if you encounter these viruses very young in life, you're very likely to have quite a different, sometimes asymptomatic or common cold experience, whereas people experiencing them later in life have much more inflammatory potential and much more potential for the serious symptoms that you normally associate with those diseases. I'm gonna look on my list here, and I have a question for Mandy Lewis. What aspects of human culture, the way we interact in our society, could affect the vaginal microbiome of women?
Well, I would expect that there are likely to be many human behaviors that could influence the vaginal microbiome, including sexual behaviors, family planning, menstruation, so-called hygienic behaviors, as well as other intravaginal and genital cutting practices that happen in different places. So, potential mechanisms by which those behaviors might change the microbiome haven't really been extensively studied, but it does appear, that, from what we know, sex partners seem to share urogenital microbiomes. The microbiome can change, microbiome changes can be triggered by menses and also the use of hormonal contraception. And then we also know that vaginal washing has been linked with higher risk types of vaginal microbiomes, possibly by eliminating beneficial bacteria, but this requires more study. And so I think, one can imagine a number of different ways that these kinds of practices could be beneficial or pathologic in the setting of human evolution.
Thank you, Mandy. So the next question is for Sujan, and it's from Jason, a CARTA student, and he asks, in much the same way that malaria has indirectly resulted in phenotypic changes in humans, e.g. sickle cell and thalassemia, has the competition between dengue and Zika produced any adjunct phenotypic changes in humans, deleterious or otherwise? And I would like to add, are there other examples of viruses that have left an imprint on the human genome?
Yes, that's a really great question from both of you. One of the most interesting and very understudied aspect of dengue infection in humans is these viruses, they circulate in Africa, but we don't really hear about dengue being a major problem in Africa. In fact, all 4 serotypes, Zika, all these different flaviviruses are co-circulating, and usually this co-circulation of multiple different dengue serotypes, multiple different flaviviruses is not a good outcome in terms of how that impacts dengue disease severity. And based on a couple of anecdotal studies done in Cuba, it seems that the African genetic diversity perhaps may be protecting those individuals from coming down with a severe form of dengue infection. And whereas in the case of people in Asia and people in the Americas, both North and South, people can come down with severe dengue. So in terms of how these interactions between the viruses and the human host, that is a really important question, vastly understudied. And I think that really calls to people in the field to start studying these flavivirus infections in Africa. And, that's a huge understudied topic.
Yeah. Can I just follow up and ask, is part of this just it just being understudied? Because they just say you have a fever and they just assume it's malaria. And
yes, so just like exactly the same issues that we're facing in terms of the healthcare system of a given country determines what kind of case numbers you're seeing with COVID. It's the same thing in the case of flavies. For Zika, in the case of all four serotypes of dengue in Africa, we don't really know the true incidence of both the mild and the severe consequences of these viral infections. And it really, again, it's the unfortunate reality of the world we live in, is this different types of healthcare system infrastructure to be able to diagnose, to detect these infectious diseases.
Okay, I'm gonna ask the next question to Manuela. How much is known about the spread of Salmonella and whether it mirrors the out of Africa movement of ancient human populations? And are there examples of Salmonella anthroponoses, meaning like where humans infected animals with Salmonella?
So those are two very nice and interesting questions. And we are learning a little bit about how Salmonella was present in ancient humans, thanks to the advancement in genomics. For example, we are able now to isolate genomes from ancient humans. And actually this February, there was a very interesting study where they showed that they were able to isolate Salmonella from humans 6,500 years ago. And that led to the hypothesis that Salmonella might have emerged in humans with the Neolithic revolution, when humans switched basically from hunting and gathering to farming. And the idea is always that Salmonella has been spread from the animals to humans, really with the advent of farming. But something interesting that came also from this study is that, for example, animals are blamed exactly from transmitting Salmonella to people, but Salmonella in pigs has actually been found as 4,000 years ago. So this led to the hypothesis that it's actually humans who transmitted Salmonella to pigs. And yes, now pigs are sources of Salmonella, but it seems that were humans first. So some animals transmitted to humans, and then humans eventually transmitted to pigs. So I think, in general, we can consider Salmonella as the non-typhoidal Salmonella has a zoonosis because they are transmitted from humans. But at some point, we were the source of Salmonella for the pigs. So we cannot blame the pigs for giving Salmonella to us like 6,500 years ago.
Thank you. So the next question is for Sue. And the question is, what class of pathogens shaped our immune system most strongly? Viruses, bacteria, protozoan pathogens, or metazoan parasites?
Yeah, thanks. I really like this question. It's something I've thought about myself, but I'm not going to be able to give an extremely satisfying answer. I think this is something that we still cannot necessarily provide metrics on. But you can think about some of the events that have probably had the largest influence. One, from a genetic perspective, on our genomes would be founder effects. And when we've had outbreaks and pandemics of the past where there were significant magnitude of death in the human population, obviously that is going to have a founder effect and have a major impact on on our genome that we propagate. The second is the prevalence of the pathogens. And I think this is something that's more about adaptation and how our we coexist with in a more endemic way, as we clearly can see genetic. We just discussed some of them, genetic genetic alleles and changes that are maintained within the population as a consequence of the coexistence with certain pathogens. There is a little bit of evidence as well that the regions of greatest diversity in our genome, such as our major histocompatibility complex, which is the MHC, which is what is used to present the foreign antigens to our immune cells, such as our T cells, to see the pathogens, that the diversity and the high level of polymorphism in these alleles is shaped by, in some ways, by certain pathogens more than others. And I think this has been probably best seen in looking at another region of our genome, which is in our immunoglobulin genes, such as the ones that create the rearranged receptors for our T cell receptor and our B cell receptor that produce the antibodies, that these germline encoded regions of, say, regions of those variable domains, for instance, that are found in the antibodies, that the germline sequence of those already has natural affinity for certain pathogens, such as influenza. So we probably have evolved naturally moderately affinity, proteins that have moderate affinity for some pathogens that we've encountered that have some evolutionary advantage to responding to those, but obviously there's still further adaptation that must happen with the immune response for better protection. So, and then our innate immune system, which is our germline, hardwired form of defense that we evolve with, that certainly has been shaped by the pathogens. But to answer the question more specifically about which class, I don't think it's going to be a particular class that's more effective. I think it's going to come from the ways in which we've adapted to our environments and these particular events that might have happened over history that had major founder effects on our genome.
Thank you. Victor?
Okay. Well, I'm going to ask the next question. I guess it could be to the group, but I'm going to focus it first to Elizabeth, because she's equally accomplished as a geneticist, as an infectious disease specialist. The malaria pestilence has resulted in several diseases as a byproduct in regions where it's endemic, like sickle cell disease and thalassemia. Are there any pathogens outside of malaria, endemic regions, that have analogously produced byproduct diseases in which, let's say, human heterozygotes experience a protective benefit?
So, well, this was, I will start to answer it, but you might know better than me, and maybe Manuela can chime in as well. And I'm thinking of mutations, the cystic fibrosis genes, and how they may provide protection against cholera. So there is a theory out there that the cystic fibrosis allele maintains, which affects, I believe, a sodium or a chloride channel, and that this is maintained in the population because it protects against cholera. Which was a big killer of children as well. And if Manuela or Victor, if you can think of some other examples, please feel free to chime in. TB?
Well, I guess, with respect to cholera, yeah. So the CFTR is a anion pump that produces, releases chloride and bicarbonate, and I guess mutations impairing its function could reduce the massive volume of secretory diarrhea experienced there. Actually, it's very interesting that Ajit Varki and Nissi and their mouse lines that they're developing, they have humanized the sialic acid repertoire in a way that some of the toxins that are produced by enteric pathogens, including cholera toxin and toxins from Salmonella, are less severe, and that might allow us to breed those mice together with, you know, the CFTR mice and find out whether there is indeed a protection once you humanize the background. Because right now, a lot of the mice just aren't susceptible to the human pathogens for us to answer those questions. Manuela, do you have some comment?
So, yeah, I would like to just comment on the fact that I always found the genetics of resistance to malaria very fascinating. And I've been familiar with this my entire life because I'm from the island of Sardinia, where basically everybody has either a beta thalassemia trait or the other trait that is also associated with resistance to malaria, which is the G6PD deficiency. And I can tell you in every family, there is either one or the other. And I think that my island is really the best example of how the entire population has evolved to be resistant to malaria, and how the heterozygous for G6PD or beta thalassemia are so widespread. And there is one in every family, even in mine.
Yeah.
I'll mention one more thing related to a pathogen that was discussed today, group B streptococcus. There is a human mutation in a sialic receptor pair. So normally humans could be said to have both an inhibitory sialic and an activating sialic that are paired in terms of their same binding interaction with ligands on the outside, but one transmits a positive signal to the immune cell and the other a negative signal. About half of the humans have a matched pair that balances inflammation, and the other half of the humans have a mutation in the activatory one, so they only have the inhibitory sialic. And we found in screening population of patients who had premature delivery, of which group B streptococcus is an important risk factor, that there was an association of the having the hyperinflammatory phenotype with a greater risk of prematurity, and published that a while ago.
Yeah, I had one other thing that I wanted to add to that, that line of thought, and it's something that we haven't really discussed today much, which is tuberculosis. And tuberculosis, of course, is one of the big killers as well, and has had a major impact on the human genome, I think. And I don't know how well this is accepted, but you know, there is some, you know, there is a hypothesis that human skin color, especially for people that live in that you know, adapted to northern climates, part of the adaptation for a lighter skin color comes because it provides more vitamin D, and it allows you to survive better, possibly survive infection from tuberculosis. So some people believe that the light skin color may be a response to tuberculosis.
Should I ask the next question, I guess?
Yeah, go ahead.
Yeah, Elizabeth. Well, I think there was a question for Chip that I think a lot of people have asked, and of course it's been in the news, and it's about whether and wildlife and human interactions in general, and how do we know that coronavirus is developed in bats?
There's been a lot of effort to capture bats in multiple different locations and study coronaviruses and other viruses that bats carry. They evolved without a bone marrow, and they do that because they can fly better, being lighter, and that leaves them with an immune response that allows them to be a lot more tolerant of ongoing carriage of an agent without clearing it. So they are really good at being able to serve as flying flasks with quite a bit of viral replication going on, and then within them, and then opportunities for viruses to mix within an individual bat and within bat populations. They live in very large colonies. So they're really socially and immunologically set up to be an interchanger of viral genetics. And when you look in bat populations, you see a large number of variant coronaviruses that are always exchanging RNA and evolving and then periodically spilling over into other species. So they really are kind of a cauldron that periodically boils over, and we happen to be on the wrong end of it catching on. It's been going on, I'm sure, for thousands of years, and there are so many related coronaviruses in bats. It's hard to go back. Sometimes this coronavirus is a good example and say this virus came across at this point in time, because the coronaviruses in people are actually more diverse with the SARS-CoV-2 than people initially thought. So this virus didn't just kind of show up in a pangolin as people initially speculated in December of 2019. This virus has been spilling over to the human population off and on for a while, and then finally managed to catch on and spread. One of the things that has happened that's different with the wildlife populations is although these events have been going on for a long time, we've done two things that have made it easier for the viruses to play with each other. One is we put a lot of these species together in small cramped conditions that wouldn't normally be together in shedding pathogens, and allow related viruses to spread among quite a variety of species in a given location at a given point in time. We also do this by creating large populations of animals with breeding facilities and for our food supply. And finally, we've given viruses the opportunity, when a spillover does occur that might well have died out in a small village, to make its way out of the village with transportation and out of the country with air transportation, and converted the human population on the planet to basically a large mixing bowl, as opposed to a large collection of small populations that evolve within themselves and develop genetics, as we heard about Sardinia, that deal with local pathogens. And now we have to think about ourselves as a global community in which the entire population of the world might have to rapidly evolve as a pathogen comes along, which we're not able to do. And so I think our current both interaction with animals in animal husbandry, and as we began to deforest areas and move into areas where animals that we normally wouldn't come in contact with, we're in contact with more often. This is the speculation about Ebola, coupled with giving these viruses a way out of where they initially are come in contact with us with transportation, has been what's amplified things that have been going on for a long time on a much smaller scale.
Oh, actually, I have a question for Sue that's kind of just sort of my question. And it's always, the lab always gives me a hard time, and they always say I never get sick, and it might be because I have an office and they don't. And it also may be because I had two kids that went to public daycare, and I got everything. But the other question is, the other thing that I do is I always religiously get my flu vaccine. And so the question is, if you get your flu vaccine every year for 20 years, are you then more protected against all the sort of sub-strains that might be out there? And does this, you know, even though you might not have the what, you might not be vaccinated against the one that's out there for that particular flu season?
Yeah. I mean, I think logically you would think that, yeah, the more vaccines you were to encounter, you would develop a broader, you know, and assuming that the drift in the influenza virus in particular, as an example, was sufficiently expanding the different types of viral strains your body was exposed to, I would think that naturally it could allow for wider, broader immunity to various flu strains. But actually, one thing that has been seen, especially from the H1N1 swine flu epidemic that we had in 2009, 2010, was that there are some more, you know, broadly neutralizing antibodies to more conserved features of the influenza virus, like the stalk domain of the hemagglutinin, so the more stem region of it, which was found that people who had pre-existing stock antibodies. Now, whether they got them from a prior influenza infection or because of vaccines, those patients oftentimes had more, had less severe outcomes with swine flu and were able to recover more in a more healthy way. But other than studies in mice had shown that if you do give a vaccine that is against different flu strains, actually where you won't have antibody-mediated defenses to the hemagglutinin portion, that's the most immunogenic and variable portions of the virus, that you will, if you give a vaccine from one strain and then a different strain for which there wouldn't be this antibody cross-reactivity, you would have the ability to boost these antibodies to the stock domain, so the more conserved features. So I think that whether or not that's been shown reproducibly in humans across the years of giving flu vaccines, because now this is being monitored more each year, because it's a more routine way of looking at human vaccine responses. But so it's possible to develop, I think, more cross-reactive protective immunity by giving more different types of vaccines to, say, flu. So I'm always a proponent of the flu vaccine, so keep doing it.
Yeah.
Okay, well, we're coming to the end of our hour, and I'm going to throw one last question out there because it's forward-minded. And I'll direct it first to Sujan, but then others could chime in. Dengue and Zika are just two recent viruses that our species is currently facing. There must be more to come. What do we know about the next one? Do you think that understanding cross-reactivity will give insight into how to fight it before it appears, or even preventing it?
Great question. And I think all of us will probably have different ways to tackle this question. I keep on talking about dengue and Zika, but what I forgot to tell you during my talk is there are 70 known flaviviruses. And that means, think about that. We know about dengue, Zika, West Nile virus, Japanese encephalitis virus, yellow fever. These are already flaviviruses that can cause devastating severe consequences in humans. Zika emerged in 2016. And with over 70 known flaviviruses, I can name a whole slew that can potentially emerge. And so this concept of cross-reactivity, we really need to understand the rules. How do we harness the good aspects? So in the animal models in our labs, it's really easy and fun to manipulate different aspects of this cross-reactive immunity and try to dissect what gives you immunity, cross-protective immunity, what gives you this cross-protective pathogenesis. And in terms of solving this problem that relate with these viruses that share this immunological cross-reactivity, what we want to do is harness this good aspects, the cross-protective immunity, and avoid this ADE type of phenomena. And I think it is doable. We need to look at more studies using animal models and, of course, humans in the context of different flaviviruses, different coronaviruses, just like with what Chip just mentioned. There are so many coronaviruses, I think we need to think about, and same thing with different strains of influenza. So what this is telling us is we really need to understand this cross-reactivity, both from the positive side and the negative side, harness that knowledge so the world can develop a vaccine, not just against this one virus, but if we really think about a lot of these infectious diseases are impacting these resource poor countries. And immunologically, if you have both yin and the yang, and these are resource poor countries, it doesn't make sense for us to think about developing one vaccine for every single infectious disease. So we can use these good aspects and try to develop, how about a pan-flavy vaccine, so that countries in Asia, they don't have to worry about Japanese encephalitis virus, dengue and Zika, the Americas, we have to think about the yellow fever. You know, Brazil, poor Brazil, made news in 2018 and 19. They went through dengue. 2016, they went through Zika. They made news in 2018 for a yellow fever. 2019, you're not going to believe this, they had the highest ever incidence in record of severe dengue infections. So this is a vicious cycle. The world, it's not logical, it's not economical to think about one vaccine, one antiviral, one drug against one of these viruses. We really need to understand this cross-protective immunity, both the good and the bad. And that's a really good question that the person in the audience asked.
Chip, do you have anything to add to that?
Yeah, I think I agree with everything that Susan said. I think the other thing is that we should not lose sight of the fact that even though we may not know what's coming next, we know that something will come next. And we seem to forget about that every time we have one of these outbreaks. Singapore and Hong Kong learned a lot about what to do when SARS-1 rolled through, and they were much more ready for SARS-2 than we were. We watched what happened and then did nothing, and we allowed our public health responses to decay, and we don't have enough people in San Diego County Health Department to trace people with SARS-CoV-2, which is one of the reasons the virus continues to rage out of control. So we should learn from this that although we may not be smart enough to know what virus is coming next or what pathogen's coming next, we know that when one comes, we have to be prepared to deal with it. And that requires having advanced diagnostic equipment out on the front lines so we see it coming with more foresight. It requires having a broad vaccine platform so that we have vaccine platforms that can be applied to multiple different pathogens. We're obviously betting on RNA vaccines here for SARS-CoV-2 in the US. Other countries are betting on other technologies, but having those platforms in an adaptable way out there so we can bring them to the table with any pathogen is very important to do, and understanding how to use them is critical. Having an understanding that we can't stop these agents by stopping airplanes from flying or by building walls. We are now a global community, whether we like it or not, and we have to be prepared to deal with this as a global community. Even if we wall it out of a small area, these viruses will continue to spread around us and will eventually get in. So we need, as a global community, to solve the problem for the world, and that solves it for ourselves too. So that's my last polemic and obnoxious statement directed at our poor response to this one. We need to do better next time.
Okay, thank you, Chip. I think that that's fantastic, and probably couldn't find a more fitting ending note for the questions, bringing it right back to our present predicament. I personally want to thank all of the speakers, and especially the audience, for such a fascinating and educational dialogue that we've had today. And pass it over to my co-chair, Elizabeth Winsler, for the very last word.
Yep. I just, again, this has been a really fantastic set of talks. I learned a lot, and the questions from the audience were really spot on. So thank you all for joining us.